Okay, so thank you very much. So yeah, today I'm going to be talking about our research project, CRC research project, which is looking at design and evaluation of targeted biosecurity systems. And to some extent I'm presenting this, what do I point at? Yeah, on behalf of Maggie, who was the postdoc who did a substantial amount of the work on this project. So the problem that we were looking at through this whole project was about optimal surveillance design. What's the best design for a surveillance system to find something that you actually don't want to find? So there's lots of talks about different methods for detecting things, but what we're specifically focused in is the design. So it's about things like where the number of samples or traps that you use, and where you actually put those samples or traps, and also about how frequently you sample. And the project was tasked with designing general methods that could be applied across lots of different case studies. But to test that, we applied these methods to 3 case studies that had different kind of scales, as I'll explain. So the first case study we looked at was grape phylloxera. And the second one was potato cyst nematode. And the third one was fruit flies. And so we looked at these together with our partners across Australia, across the state departments, and also in New Zealand as well. So I'll talk about each one in turn. I'm aware that some of you might have seen some of these recently, but there's a few new bits in here as well. So grape phylloxera was our first one, and that's me and Maggie out in the grape blocks in Victoria, learning how hard it is to look for phylloxera, or how hard it is to find it. It's pretty easy to look for it. So based on our extensive experience out in the fields, we then built our simulation model. So the simulation model looks like this when it's running. So you can see that there's some different soil types in this model, because that was one question people were particularly interested in. So 3 different soil types, or as many different soil types as you like. Simulation runs and it shows the population densities of phylloxera building up in the field after one initial incursion at a certain point. And we can simulate high virulent situations like the one on the right, or low virulent situations like the one on the left, which might represent different environmental conditions or different genotypes of the phylloxera, or different rootstocks of the vines, for example. So you can see that the populations build up much faster on the right than the left. They spread much faster as well, but they spread slower into the low suitability soil because it's low suitability for the phylloxera. So over the top of that simulation model, then we can overlay different sampling strategies, different sampling designs. So the standard method is to go out, dig a bit of soil, look at the roots, and then we'll also hear about some other methods like DNA sampling. It doesn't really matter what the method is, it's about where you then take the sampling to apply that method is what we're interested in. So we looked at the standard sampling designs, just based on a grid, every 5th block of every 3rd row, for example. And then we looked at what would be the effect of increasing or decreasing the density of sampling. But what was particularly we were interested in was methods that target the high suitability soil, because some of the experts thought this might be a more effective way. So the same number of samples, but specifically focusing those into the high suitability soil. So in this example that's shown on this slide, you can see there's more samples in the high suitability soil and fewer samples in the medium suitability, and none at all in the low suitability soil. So we run the simulation many, many times, thousands of times, and we record for each design at what point in time does a first detection occur. And then we can record other things like the distance of spread at that time, or the spatial extent of the infestation, and so on. So the top left-hand graph here shows the initial detection, and then the y-axis shows the probability of detecting it within that number of years. So basically, further up and to the left is better. It means that you're going to have a higher probability of detecting it within a certain time. We can see that the red lines represent the virulent situation, the blue lines the avirulent situation, the non-virulent, so there's quite a difference there, but we can't control that, of course. It will depend on our environment or the phylloxera strain. But what we can control is which method we use, and the different colours within the blue or within the red represent our different strategies, like focusing more on high suitability soil, for example. And so what's the takeaway here is just that there's quite big differences in some measures between the different strategies. So to summarise those differences, the most interesting result for us, for everybody really, was that surveillance design that targets high suitability soil was considerably more efficient at finding phylloxera. I guess also interesting was that sampling density had relatively little effect, so we could cut down the sampling density without greatly reducing the efficacy, and increasing it didn't have a large benefit. And another striking result was that with low virulent genotypes in low suitability conditions, the infestation could be there for a long time, even up to 100 years before it was actually going to be detected. So that's just a bit of a warning. So the next case study was potato cyst nematode, and here again we focused on Victoria. So we got data on the potato growing areas of Victoria, the statistical areas, and then the number of properties, different kinds of properties within those areas, and divided those, or they were divided into fresh potato production and seed production. And then our model simulated movement of PCN between those different regions and also among the properties within those different regions. So again, we created this simulation model. Now it's at the scale of the whole state instead of a field. And in the simulation that's running there on the right, you can see the green and the black represent the number of fields of seed and fresh potato. And when they become infested, they turn yellow, just to show that they're infested. The size of the yellow can represent the number of properties within that area. And in this one, there's no surveillance or quarantine whatsoever. So this is what would happen if we just did nothing. You can see that within 50 years, it's spread across the whole state pretty successfully. There's no detection, there's no blue in this one. So if we add some surveillance now, what happens is that they turn yellow when they get infested briefly, and then reasonably quickly with this one, the detection occurs, so we know they're now infested, quarantine is applied, and the overall spread is much slower, so that slows down the spread. So we wanted to predict the spread under different surveillance strategies, assuming that the quarantine strategies were fixed. And so again, we did lots and lots of simulations to get a good profile of the different risk, the different distribution of possible results. And we get results like the one on the left there. So again, up and to the left is better. Now the line that's to the far right, so that blue line is the baseline. That's the current surveillance, which is only on seed potato, a certain proportion of seed potato, I believe. And then all of the other lines are saying, well, what if we did a bit of extra surveillance? What would happen? And so across the bottom, the x-axis is the number of properties infected after 50 years, and then there's probability showing up the side. So basically any kind of surveillance strategy shifts it across to the left, improves results. But these lines are all for exactly the same amount of sampling effort, and the only difference between them is where we apply that surveillance. So at least for the scientific purpose, the academic purpose, the question was, with a given amount of effort, what's the best way to use that to slow down the spread? And we can see there's a big difference between those different strategies. The government people that we talked to were also interested in generating risk maps, and so the maps there on the right show different risk maps for whether different locations are likely to be sources of ongoing infestation over the next 50 years, or recipients become infested over the next 50 years. So they're under different assumptions from the left to the right, different dispersal assumptions. I wouldn't count on those too absolutely, but the possibility is there to use this model to generate those kind of risk maps. Okay, so summarising those kind of results. So survey density in this case made quite a big difference. A little bit of extra effort really slows down spread over the next 50 years. So there's a big incentive to try and do some extra surveillance if that can happen. There was a big variation between different strategies. So different survey arrangement with a fixed density or a fixed number of samples each year made a big difference. And the strategies that were generally the best were this kind of strategy where when you have a detection at a given location or within a given locality, so there's a whole lot of properties within that locality, you have a detection. The surveillance strategy that tended to be the best was one that mixed a regional scale surveillance, so surveillance at a bunch of the properties within that location with a little bit extra, a little bit of bet hedging of random surveillance out beyond that. So maybe not surprising, but we thought of all kinds of sophisticated options that might be better. None of them were actually better than that one. Okay, so fruit fly was our third case study. And for that one, we built another simulation model. So it's a separate model for each one, but I guess the ideas and the technologies were transferable between these different models. So this one is at a sort of intermediate scale, rather than the state scale or a field scale. Now we're looking at the scale of a small town. We're sort of motivated by the idea of maybe Kununurra or a town in the north of Australia. The town has orchards around it, but it also has individual fruit trees scattered through the town, and they will be at different densities depending on how old a suburb is, whether it's residential or commercial. And we know that there's high-risk introduction sites. For example, these could be caravan parks where travellers are likely to dump their fruit as they come through from Queensland, bringing Qfly potentially in with them. Or they could be shopping centres, they could be visitor centres, those kind of places that anecdotally we know are higher risk. So we simulated initial incursion happening that's likely to occur near one of these sites, but not necessarily. It could potentially occur anywhere. And then that invasion unfolds, spread happens, populations build up. Of course, it all depends. Once we run the model again, it'll be a different initial incursion site, and the invasion will occur differently each time. So we have to run this model again thousands of times to get a good risk profile. And then again, we can overlay different surveillance trapping designs in this case. There's designs for where we put our surveillance traps. So the default, the standard is a grid-based design. We can say, well, how does that compare to random? And then we can design a whole bunch of strategies that we think might outperform that grid. So the one at the top there is one that sort of targets the high-risk introduction sites first. The one on the bottom is one that spreads out the traps so that there's one trap for each number of trees, so you end up with a lot in the orchards. And the one on the bottom right is just an ad hoc design that was tried to really target high introduction sites but protect the orchards as well. So any design that you like, any design you can imagine. And I run this as an exercise with my students as well, as a training exercise, and for them the project is about trying to come up with what they think is the best design. So they get to be creative. So results, because I did this slide instead of Maggie, it's flipped around and now down is better. So just the y-axis is flipped around. It's also on a log scale, so some of those differences are much bigger than they probably look. And what we see is the black line is the default grid surveillance design, which is sort of in the middle there. So we can do a lot worse. Random, for example, is considerably worse. But we can also do considerably better, like that green one is that first strategy. And it's much, much better, like up to 10 times more likely to detect or 10 times less likely to be undetected after a given amount of time. So similar, we can say, okay, that's days to detection, or we can look at the number of trees at first detection, whatever other measure we'd like to look at and get similar kind of results. So we went on and did a detailed analysis of trade-offs about, for example, whether we cluster surveillance more and more around the high introduction sites or spread it out to hedge our bets a little bit more. and tried to work out exactly which was the optimal design for that. And we came up with exactly, for at least one particular scenario, a method that would work out the optimal design based on any given number of traps in total. So that one was for an Australian case study, someplace northern Australia, and it was sort of based around medfly. The model was parameterised for medfly. So then we turned our attention to New Zealand, together with our New Zealand partners, to look at a specific case study that they had. So the aim here was to analyse the confidence that the surveillance they'd done in response to an incursion, in order to follow up on an eradication effort, would be successful in detecting any pests that were missed during their eradication efforts, and also to identify potential improvements that they could use in future. So this case study we looked at was a Qfly incursion in Auckland in 2015. You can see our fly there in the middle of the picture. So their response basically involves setting up a high intensity management zone, 200 metres, and a medium intensity zone up to 1.5 kilometres around that initial detection. And that involved a very high density of traps in the middle zone, and then a medium density of traps in that other zone, and then the normal density of traps out beyond that. It also involved lots of public education, not moving any fruit, making sure that anything went into bins. So the idea was that there's probably, if we found one fly, there's probably a whole bunch more. There's probably some fruit that's infested as well with larvae. So if we can get rid of the fruit, trap the flies, we hopefully get rid of the larvae and get rid of the adults as well. But unfortunately, it's quite likely that we'll also miss one or two. And so if they then show up in traps, we extend the high density zone around where those further detections occur, and we might find more out there as well, and then we extend those out and so on. So that's the method that we were basing it on. So our method was to simulate the initial incursion and the detection, and again, do that many, many, many times to get a full risk profile. So luckily they had the data on the location of every single tomato plant in that area and everything else that was a potential host with typical New Zealand efficiency. So we then simulated, after the initial detection, we simulated their eradication efforts exactly as they'd done it, and generally these removed most Qfly. Then any individual spread, the eradication and surveillance continues. That's all simulated. And then we did that for what they actually did, but then we did it for a whole lot of other alternative strategies as well. Okay, so the strategies that we looked at here was the baseline, what they had actually done, and some alternatives like changing the distance of the initial high intensity management zone. So shrinking it to 150 metres or extending it out to 250 metres instead of 200. Changing the trap density, so it wasn't every single tomato plant that had a trap, to 75% or 50%. And again, we get results. So this was Maggie again, so it's going up and to the left is better. So the original shown in black here. And we can see again, it's as we'd expect. If we increase the distance of the high intensity zone, we do better. If we shrink it, we do worse, change the density. But probably most important is how often the eradication was successful. So is that 2 minutes to go? Yeah. Okay, so we can see the percentage of simulations out of 1,000 different runs here where eradication was successful. So the original 100%, in all 1,000 it was successful. So it gives us a confidence that what they actually did was pretty safe. If we shrink the distance a little bit, 2 of the 1,000 were not successful. If we increase the distance, it's back to 100%. And if we change the density of the traps, then we start to actually have a reasonable chance that it would be unsuccessful, like 1% of the time or 4% of the time, and it wouldn't be. So it kind of indicates that they did pretty much exactly the right thing, that anything less would start to have an increase of risk, but anything more doesn't give much benefit. Okay, and the numbers they had matched. The numbers that our model produced in terms of number of traps and things pretty much matched exactly what the number of traps they'd used, so that gave us confidence in the model as well. So conclusions, the number of traps can be reduced, but so is the confidence. Survey frequency can probably be reduced. And the focus on prevention is important, that it's a big, expensive effort, and this model shows that you can't do any better, you can't save any money on it, so it just emphasises again how important protection at the border is to stop these kind of incursions happening at all. So I'll just quickly show this. So this is, I guess, for all of these models, it shows how a model can integrate our biological understanding to look at management questions. So we have information about biology, such as fecundity and dispersal. We have lure distance, and we have spatial data. We propose a surveillance design, and the model then tells us a predicted outcome for what would happen. We can then use that predicted outcome to refine our surveillance design. If we do that many times, we can come up with recommendations. We can go back and say, well, what if the biology was different? What if we're not confident, or what if it was a different species? How does that change the recommendations? The same with spatial data. What if we're not sure about where the trees are? Does it change things or not? Or if it's a different location? Or a question like, if some bright person here comes up with a lure that has twice the distance, how does that change the outcomes? How does that change the overall surveillance efficacy? And then we can ask questions like, well, these are recommendations, but how well are they going to fit with politics? How well will they fit with simple logistics? And also, how does the economics stack up? For sure, having twice as many traps is going to be more effective, but is it worth the cost? And therefore, we can get rid of these pests that we don't want. Okay, so thanks to all of the partners, of course. Work wouldn't have happened without them, and we'll hear from some of them now. And that's the email if you want more information.